Okay, well, thank you for inviting me. I guess beauty is in the eye of the beholder. I kind of think about looking at the tumor microenvironment by multiplex IF or IHC, like the Jackson Pollock to the left. Whereas genomics is, very clean and predictable, like the Damien Hirst on the right, which looks a lot like a circus plot. So maybe David prefers Jackson Pollock over Damien Hirst. So anyways. I think the topics for today are really, to talk about TMB as a biomarker. And certainly David alluded to, how complicated it can be. What mutations do we measure? How do we define a threshold? How do we think about it in the context of PD-L1 and whether we can apply it to all checkpoint inhibitors as well as pan-cancer? So certainly when we first started using immune checkpoint blockade, it's been about 10 years ago now, and we saw the lung cancer patients that we treated. The responders were in the patients who had a prior smoking history and not in those that were never smokers with lung cancer. And I think that since then, you can see most of the curve to the right has some sort of FDA approval for the treatment for those cancers, whereas that to the left, not as much so. And then this nice sort of article review that was published a few months back in the New England Journal shows the relationship between response and the number of tumor mutations. And I think the poster child for TMB is the mismatch repair deficient tumors, both colorectal and non-colorectal, which are associated with not hundreds but thousands of mutations having a very high level of response to immune checkpoint blockade. And these tumors, as you know, are now, we have FDA approval based on pan-cancer approval in this tumor based on a high tumor mutation burden. I think that before we sort of talk about TMB, I guess an important question is why does TMB predict for response to these therapies? And the sort of pie chart to the left shows that it's. I think we need to think about the type of mutation that is within the tumor, the type of mutation that leads to a change in the amino acid, so the immune system can recognize this as a foreign or non-self protein. And so when we think about TMB, we don't typically count the silence mutations and probably not the nonsense mutations because there's less of them. And it's really the missense mutations and the indels, which are considered to be immunogenic, and changing the amino acid in such a way where you lead to very high affinity binding to the MHC as well as the T cell receptor. That change that in protein binding is one that is critically important in initially inciting the immune system, but ultimately leading to adaptive immune escape. And what we're doing with immune checkpoint therapies is restoring T cell recognition to these highly immunogenic neoantigens. When we think about TMB and we think about counting mutations, we've used whole exome sequencing historically, and I think that's been challenging logistically, financially to do whole exome sequencing on all of our patients. There's now increasing data looking at correlation of targeted panel data, such as this Foundation Medicine analysis showing a good correlation between targeted panel with whole exome sequencing. So I think that we to some extent can move away from targeted panels for clinical trials in a practical way. But keep in mind that the targeted panels don't give you all the information that you need. They don't tell you about germline HLA. They don't tell you about somatic HLA loss. They don't tell you about tumor heterogeneity. So I think in some ways it's moving the field forward, but it's also moving the field back as we try to create some sort of a composite genetic nomogram for who will respond based on genetics. So I think it's good and bad both. And this is just a summary of what we do when we do whole exome versus the two targeted panels where there are some validated data. When we do whole exome sequencing, it's basically 30 megabases versus sampling of that 30 megabases with in the range of 1 megabase with Foundation Medicine versus NGS. Other people are trying to bring down the number, but there probably is some threshold that will reach where you can't go below and accurately recapitulate the exome data. People get a little bit confused about the number you get from Foundation Medicine versus whole exome sequencing because if Foundation Medicine is 1 megabases and whole exome is 30 megabases, you should just be able to multiply the number from Foundation by 30 and get the exome data. But the reality is that their algorithm is based on counting both synonymous and non-synonymous mutations, so you need to factor that as in a conversion. Also, when you use a targeted panel, you're estimating the mutation without germline matched normal data. You're estimating it based on algorithms and using private and public databases on what the sequence should be. The MSK impact panel is a little bit different, where they are only counting the somatic missense mutations using patient-matched normals, and so you'll get a little different number from that than the Foundation Medicine. But you can, in general, do a more straight conversion than you can with the Foundation assay. So the complexity is that everyone else that does these platforms is developing their own platform for TMB estimation, and how do we kind of align all these efforts? And one effort that's being pursued right now that is through Friends of Cancer Research, where test developers that have their platform, as well as pharma companies, FDA, NCI, Academia, are working to create some sort of a standardization in terms of thinking about this data using TCGA data as well as cell line data to create a reference whole exome standard that can be provided to the individual testers to create some sort of alignment in terms of how we use this threshold. This is the tough part: is what is the TMB threshold? What is the number that you need to be? What's the equivalent of PD-L1 more than 50% for lung cancer? And it's not straightforward. When we think about the phase 2 data, remember you're thinking about just the difference in outcomes between the TMB high versus TMB low tumors. You're not comparing it to the standard of care. So you've got two ways of looking at this data, sort of comparing high versus low, and then comparing the high versus whatever the standard of care is in that patient population. But this is most of the data that we have so far. And I think for lung, for bladder, and maybe for head and neck cancer, it all seems to be coming out around 200 missense mutations, or 10 mutations per megabase by Foundation, or 7.5 mutations per megabase by MSK. This data is definitely colored by PD-L1 expression. You cannot, I don't think about TMB without PD-L1. If you have TMB and some PD-L1 expression, the differences are more profound than if you have TMB without PD-L1 expression. So I'm certainly not suggesting that TMB can be thought about sort of siloed from other parameters such as TMB or gene expression profiling. So everyone's familiar with CheckMate 026, which was a phase 3 randomized trial comparing nivolumab with chemotherapy first line in lung cancer in patients who are PD-L1 more than 5%. This trial was a negative trial. And the investigators in this trial did go back and do a whole exome sequencing and looked at TMB in this trial. In the high TMB group patients, which was defined in the upper tertile, which is over about 240 mutations, those patients performed better than chemotherapy with a PFS of 10 versus 6 months. To David's point, there is no overall survival benefit from this trial, so I think that this retrospective analysis needs to be looked at more prospectively. And I think that the curves on the right show that PD-L1 is important. So if you have high TMB with high PD-L1, it's kind of a bit of a home run where your benefit's going to be massive, whereas if you have lower levels of PD-L1, the benefit seemed to be less dramatic. I think to me the most interesting parts of the curve were in those patients who had low or medium TMB, and those patients, irrespective of PD-L1 expression, did very poorly. So I think maybe David would agree with me that if you have no PD-L1 and if you have low TMB, chances are that you're not going to respond to immunotherapy. So when we think about our clinical trials that we're designing, we talked about some of the clinical trial designs quite a bit earlier this morning. I think that you have to have TMB as part of your analysis to understand or make sense of the data that you're looking at. The IMvigor 211 was a phase 3 trial in urothelial carcinoma comparing atezolizumab in platinum pretreated patients to atezolizumab or chemotherapy. It was a negative trial based on its primary endpoint, but if you look at those tumors who had TMB high, and this is overall survival data, the hazard ratio was 0.68. And if you looked at those tumors that were TMB high and had some level of PD-L1, sort of 2-3, IC2 or 3, the hazard ratio was a very strong 0.5. So there definitely is a story here where it is important, particularly when you do these large phase 3 trials. 227 was an important trial because it really was the first trial to prospectively look at TMB as an endpoint in a phase 3 setting. So although the trial fully enrolled before the primary endpoint was changed, nevertheless, it was a prospective evaluation where the subset of patients who received nivolumab and ipilimumab were compared to chemotherapy prospectively with a TMB threshold of 10 mutations per megabase that was established in previous phase 1 and 2 data. What was also interesting that is when you gave the combination of CTLA-4 and PD-1 therapy, you actually didn't lower the threshold, which I think was what a lot of people, myself included, thought, well, maybe we can get more response to a lower level of TMB threshold, but that wasn't the case. It remained at around 10 mutations per megabase, whether you gave single agent PD-1 or combination with CTLA-4 therapy. And in this trial that was published recently in New England Journal, showed that in those tumors that were more than 10 mutations per megabase, which was not an insignificant number, it was about 40% of patients, the PFS was 7.2 versus 5.4 months, and the 1-year PFS was resoundingly better than with chemotherapy. What was also interesting is that unlike with single-agent PD-1, PD-L1 therapy, where benefit with TMB was required some level of PD-L1 expression, the benefit with combination PD-1 CTLA-4 was irrespective of PD-L1. So whether you had no PD-L1 expression or high PD-L1 expression, if you had high TMB, the benefits were similar. And this was similarly observed in a small cell lung cancer trial. It was not a phase 3 trial, but if you look at small cell where PD-L1 expression did not, at least tumoral PD-L1 expression did not correlate well with outcome, you saw the immunotherapy combination doing very well in both PD-L1 positive and negative tumors, and with a high 1-year survival with combination CTLA-4 in the high TMB population. So I think we'd love to say, okay, we can just use the 10 mutations per megabase, treat every tumor that's over 10 mutations per megabase, and we're good to go. But I think that's a little bit simplistic. I think that the benefit is going to be very tumor specific, and we definitely have tumors which behave very differently. Renal cell carcinoma is a prime example where these tumors don't have high TMB but respond very well to immune checkpoint blockade. They do tend to have a more different profile where they tend to have more insertions and deletions, and these indels lead to frameshift mutations that lead to a long reading frame, so you can generate a large number of epitopes, neoepitopes in renal cell carcinoma. So this is a little bit different and doesn't really rely on TMB. Merkel cell is not very mutated and responds, doesn't require TMB. And similarly, mesothelioma has very few mutations, yet we see clear activity in mesothelioma. Last couple of minutes or minute, just really talking about the chemotherapy combinations, which have permeated our lung cancer world in a large way. Is TMB relevant to chemotherapy combination? Well, I think the answer is maybe. Some of the data that was presented at ASCO this year looked at, again, the same 227 trial, but there was a nivolumab chemotherapy cohort in the patient population that were less than 1% PD-L1 expression. And if you look at the outcomes in patients that got Nivo plus Ipi versus Nivo, chemo versus chemo, in the more than 10 mutations per megabase, definitely the nivolumab in chemotherapy was better than chemotherapy, but the data did, in a non-statistical way favor the immunotherapy combination. But if you had less than 10 mutations per megabase, the curve on the right, it didn't seem like it helped at all over chemotherapy. So when we invest these large amounts of resources with chemotherapy and immunotherapy together without really having any idea who we're helping, I think we really need to think about this data, both from a toxicity perspective of both patient-wise and financial, and think about this a little bit more carefully. What was also interesting was that the durability was much less, even in the patients that had high TMB, with those that got nivolumab and chemotherapy had less durable benefit than those that received immunotherapy combination. So ipilimumab may well be important for memory effects and leading to ongoing immune surveillance that's being reset by these therapies. Finally, I think that we need to learn more. I think that we and others have looked at tumor heterogeneity as well as somatic and germline HLA, and just for time reasons, we won't go into that. But I think that there's more for us to learn. I think TMB is a work in progress. And in conclusion, I think it's a valuable biomarker. It's been an effective patient selection tool in phase 3 trials. The targeted NGS panels do correlate with exome data, but only give a piece of the information. Threshold may vary by tumor type, so I think we need to learn more about all other cancer types. PD-L1 expression may be important, and I think that platform validation is really going to be important with all these other assays that are coming on board. Thank you.